you heard from Dr. Frau at that last lecture, because as the title says, it's Gene Expression Part 2. So it's going to be a little bit more complicated. I'll try to go slow through it. We've also pared down all the many things that can happen so that you only have about one example per idea or concept that we talk about today. There are many, many examples for each concept. Okay? We'll only talk about one particular example. So the first idea that we need to think about is that cells have memory mechanisms. The collection of molecules that are enclosed by membranes, which we call cells, they can remember across cell division. So we'll talk about some basic memory mechanisms, and that's absolutely crucial for you having the same kind of cell being made again and again, and the cell persisting to be the same across time. The second concept that we'll talk about is that genes and gene regulatory proteins can be wired together as little devices inside cells. And these devices can be combined in logic that can get exceedingly complex, much like if you looked at a printed circuit board or something that's a chip inside your phone. It looks incredibly complex. It can be that complex in the way the molecules are wired together. In fact, it's more complex than that. So we'll talk about how that happens and what are some ways in which you can put together gene regulatory proteins and DNA to get behavior that is not simple. Okay? And there are some extraordinary things that eukaryotes can do with the DNA information that's inside the cell. One of the most exciting things that are still being pursued actively and not yet fully understood are that eukaryotic cells can pick one of the two copies of DNA for making protein, which means we get our maternal copy and a paternal copy that's present in each of our cells, but the protein that's getting made can be made from just one of those copies. So there are mechanisms that select for that, and we'll talk about two such mechanisms, one called imprinting and another called X inactivation. You probably have heard a little bit about these two things, but we'll talk in greater detail about how these mechanisms come to be. And as Dr. Frau mentioned in his last lecture, the first step of gene expression is where you decide to make mRNA for a particular protein. And he talked about the many regulatory steps, ways in which just that first regulatory step could be regulated. After that, all the way out to making the protein, every single step can be regulated. You can think of it as elongation, which is where the polymerase, once it decides to bind to the DNA and makes RNA, how fast it elongates, is it moving along and making RNA? Splicing, which is where introns get removed and you have the exons put together to make protein. 3' end formation. This is where you have a long piece of RNA that's made by the polymerase, and then you have to make a specific endpoint at the 3' end of that long RNA. That's under regulation. And editing, which is an odd thing. So far you've heard about the information being coded from DNA, then in RNA, and then you get protein, central dogma. Well, there's something that can happen at the RNA level where the information that's coded can change through a mechanism that's evolved called editing. We'll talk about that, how RNA gets edited. And nuclear export. This edited RNA has to get out into the cytoplasm, which is where, or the cytosol, which is where translation happens. So that step can be regulated. And within the cytoplasm, it can be regulated where it's localized. Maybe it's localized at a place where you can have high rates of translation, or it's localized at a place where you don't. So we'll talk about that as well. And the axis of the ribosome on the RNA, how easily does the ribosome get on the RNA and make the protein? And finally, how quickly is the RNA degraded? So just because you make a lot of RNA does not mean you will get a lot of protein. I hope Dr. Frau mentioned that everything that you see at the RNA level doesn't need to match up exactly at the protein level, even though you have a whole mix of RNAs and a whole mix of proteins that are getting made. So it's again going to be organized, this lecture, in these 4 steps. I'll pause at each step and hope that everyone's got what's happening. So the first step, which is the simplest of things to think about, is how could a cell remember something? And the easiest way to imagine that is, let's say there is one gene from which a protein A, gets made. Then the simplest thing that's necessary to remember is to have the protein persist across a cell division, and then the protein activate itself. So that's what's depicted here. Let's say there's some signal that turns on this protein, a cell, let's say you're shining light on it, whatever, depends on the cell. If that protein A is made and the protein binds to itself, the gene regulatory region on itself, to activate more production of A, you can imagine when you have protein A in the cytoplasm, you divide, there's still protein A, it keeps going. Okay? This form of memory mechanism is very simple, and it's called feed-forward memory. Okay? Positive feedback or feed-forward memory. And you can see that depicted here as the simplest of mechanisms. It's a positive feedback loop, meaning once you make a protein, you feed back on the gene and say make more. Okay? That's all that's needed as a mechanism to remember. Now, you can imagine just this principle can occur at many different levels. Okay? So there are 4 different ways of remembering we can think of. A second one, first was just make the protein that binds the DNA and just have more of that. That's it. The second one would be at the level of histone regulation. We've already talked about how histones are crucial in eukaryotes for deciding whether a gene gets expressed or not. You can imagine if a cell has active chromatin, some modification step happens that makes the chromatin now inactive. Then because there are mechanisms that can perpetuate all the chromatin marks across DNA replication, across cell division, you can have that new chromatin state just be remembered across. Okay? A third way is through remembering DNA methylation. This is probably new to you. We will talk a little bit more about this later in the lecture today. But DNA methylation refers to the methylation of cytosine on the 5th position on the base. And that methylation changes how gene regulation happens. And if such DNA methylation occurs, there are ways to remember the methylated state across division again, which is another way of remembering this changed new state. Okay? And a final one, which unfortunately we're not going to talk too much more in detail except for this one slide, is remembering protein conformation. And that's, how many of you have heard of prions? A few, okay. So these are protein conformers which just look different from the native protein, but they have the ability to convert other native proteins to look like them. So for example, let's say a normally folded protein looks like this, spheres. If there is a conformation change in the protein, no sequence is changing at all, just the folding changes, it now forms some kind of template on which you can have many more proteins that are also misfolded, like these sheets. So if these sheets now persist across cell division, doesn't matter what the DNA is. The DNA sequence can be exactly the same. But because these sheets persist across cell division, they will force the other proteins to take this protein aggregate state. So this is another way in which you have memory, and this is a form of memory that doesn't care what the DNA is, right? Because it's the protein itself that's remembering what's going on. Okay, so any questions so far on memory mechanisms per se? Good. They're just simple ideas, but you're just remembering what happened in one cell to the next cell division through these 4 different ways. Now, just because you have a protein that can act on itself doesn't mean the outcome will always be the same. Exactly how the protein acts on itself will dictate what sort of outcomes you get. And some of this might be familiar to people if you think about electronic circuits. And you can imagine diffusing proteins that are interacting with each other from the same conceptual standpoint. So here's the first idea, positive feedback loop, which we've already talked about, where protein A, schematized here with this yellow box, acts on itself in the schematic to increase its concentration. Now what's shown in the schematic can be something like protein A goes back and binds on the DNA, makes more of itself, or let's say protein A is being degraded a lot, it phosphorylates itself, making it more stable, and every time it's phosphorylated, that can phosphorylate itself again and keep being stable. So any way in which the protein acts on itself to increase its concentration would be a positive feedback loop. A second one is where a protein acts to inhibit itself. Now that seems counterintuitive why you would have such a thing in cells, but imagine if when a protein is made, you want to not just make it, but keep it at a certain concentration in the cell. The way that would be achieved is you have higher and higher amounts of protein, leading to an accumulation beyond a level where now at this very high level, the protein will bind and inhibit its production. So anytime it crosses that level, protein production stops, right? So what you have is a negative feedback loop, which is used to maintain proteins at a certain concentration. And the final one, this is also very common as a regulatory motif, if you will, in cells, where you have one protein inhibiting another, and the second protein inhibiting the first. And this is a very versatile and sort of diverse outcome. Outcomes can occur from this particular arrangement, where it's called a flip-flop device, and as the name suggests, that's what happens. You can imagine if protein A goes up, it inhibits B a lot, and because it inhibits B, the inhibition of A by B is no longer there. Now A starts going up again, okay? So you could oscillate back and forth between the levels of A being high or the levels of B being high, and it all depends on what the quantitative details are. We'll talk in detail about one particular kind of circuit, which is the deliberate oscillation over time, which cells achieve and use. The feed-forward loops can also help cells distinguish between what is a signal and what is noise. And as we talked about already, a membrane-bound collection of molecules, that is the cell, needs to interact with the environment. There's something on the outside, and many things on the outside are necessary for the protein, for the cell to even live, right? They have to be growth factors and so on the outside. But how does the cell know if something transient just happened, and that was just noise, or if there's a genuine signal? So it does what you can think of as kind of integration, where you have an input that acts on a protein, which then acts to generate an output. Let's say this would be making more of this protein Z. But that does not happen unless enough of A is accumulated to also activate B, and B has also accumulated to then activate Z, okay? So only a sustained signal will allow enough of A and B to be made so that now Z gets made. So here's that schematized. If you have a transient input in time for a very short time, then you don't have output. But if you have a sustained input for a long time, the output is okay. Now those are just sort of the basic ways in which we can think about it. There are many more complicated circuit arrangements that are there. I want you to think of this clearly as the way in which we try to understand what the cell does. Still remember that within the cells there are just molecules that are bumping about and interacting with other molecules, etc. But we try to understand what's happening in the cell, or what is the cell computing, by putting them together in these forms of circuits. And we don't quite know exactly how any whole cell computes, okay? So here's an example of an early developmental circuit that was worked out over years of work. I think Eric Davidson was one of the guys who pioneered this. There are, let's say, maternal and early signals, say proteins or whatever that's given by the mother into the egg. And then there is development of three key germ layers that happens. There's mesoderm is in the middle, endoderm is in the inside, ectoderm is on the outside. So here's, let's say, you're making mesoderm and endoderm, and there's some machinery, which is what we think of as everything that's in the cell, that makes sure that you get one or the other. And if you really look at it, that's how it looks. So you can be sure that's not in your exams. Nobody's going to ask you, can you draw this thing? But it gives you an idea of the level of complexity that exists in a cell. And so everything that we're learning are sort of bits and pieces of what's going on in the cell to give us an idea of the ways in which a cell computes. Okay? We still don't know how the whole cell computes at all. But using these sorts of steps hierarchically, one by one, we can begin to figure out how this miraculous process of development happens. You start with that one cell embryo that's going to give rise to everything. How do you get that? Do you code for every single cell type's identity? That's not the case. You end up having combinatorial regulation, which means you begin with just, let's say, 5 protein types. Then you can get, by combinations, many, many, many more. What's illustrated here is with having just 3 proteins, how you end up getting 8 different cell types. So let's say there's an embryonic cell, and if a gene regulatory protein 1 gets activated, but it's activated in only one of these 2 cells, those 2 are already differentiated. So that's this process of differentiation where you go from something that's homogeneous in the beginning to something that's now different from each other. Okay? And subsequently, if you now both were induced to make, let's say, protein 2 and protein 3, they would still be different from each other. Because on the left, you'll have only 2 or 3. On the right, you have 2 or 3 in combination with 1. Okay? So the concept here is, if I were to ask you, how could cells generate so many different types in an organism, then your answer might be it uses combinatorial logic of many different genes being expressed in different subsets of cells. But the way we figured this out really, okay, why do we think that this sort of thing is happening? The way we figured this out is because we stumbled upon, during the study of organisms, we stumbled upon genes that are master control genes. They are such that if you only express that one gene regulatory protein, you can completely transform the fate of a cell. So imagine the cell has millions of things in it, many, many gene regulatory proteins in it. So let's say from your skin, you could isolate fibroblasts. We'll talk about what those are later. It's just a bunch of skin cells, let's say. You can then put in one regulatory protein called MyoD, myo for muscle, such that it now transforms this cell into a muscle. And muscle cells are dramatically different from skin cells. Some of them are binucleus, meaning they have to fuse and you have multiple nuclei inside muscle fibers. That's how it is in us. And you can see here, shown in this early experiment, in orange are nuclei. Here's one cell in which there are 2 different nuclei. And that was skin cells into which this particular protein, MyoD, was put in. You don't have to worry about all these gene names, but I want you to get a sense of what the organization looks like. You have in blue some signal from the outside. Remember that integration arrangement? These class of proteins will directly give you muscle structure genes, but they would also need this second protein. So this is like A and B together giving you the Z genes. And in green are the feedback loops that are positive feedback loops, so that once you start with MyoD, you keep going making more muscles. Okay? So just MyoD expression in chicken skin cells was sufficient to make them into muscles. Even more dramatic changes can happen if you take the correct gene regulatory protein, but then you express it at a different place at about the right time during development. This was alluded to, I think, in Dr. Frauett's lecture when he was talking about homeobox proteins, because they lead to homeotic transformations. And these are some of the craziest kind of biological results that I learned as an early student. So here's a fly experiment where if you, let's say, have a group of cells that give rise to the eye, and they have a gene regulatory protein called EY here. It's called eyeless. We'll talk about that in the next slide. You took those cells and moved them around in into a different place, or you took that protein and expressed it now in this different place, slightly posterior, in a set of cells that typically, let's say, give rise to the leg. Then you get this crazy transformation where now there is a fly eye on the fly leg. Okay? And that's shown here. Here's the actual leg with the fly eye. So what this is teaching us, in addition to it being really a bizarre and gross experiment, is that things are put together in a modular fashion. So remember that MyoD experiment, you put in the MyoD protein and immediately you get muscle out of the skin. If you put in this eyeless protein, you're also generating the eye wherever you put it in. And Drosophila guys like to name their genes in really funny ways, but so all of these things, toy is twin of eyeless, EY is eyeless, Achilles, and then eye is absent, but at some point you run out of eye-related names, so Dax is Dachshund. You don't need to know these names, but again, look at the architecture of how these genes are put together. In blue is what's happening as you go down, and in green you have the same positive feedback loop. So that's why just expressing eyeless somewhere is sufficient for you to now drive eye generation there. And the naming is a little bit odd because eyeless is what happens when you don't have it. So that protein is named eyeless because that's what happens when you don't have it. So if you took that protein, expressed it somewhere, you get an eye there because of positive feedback loops. Okay. So, have any of you been jet lagged? Quite a few. Do you know how to fix it? So, there is now active research going on trying to see how you might fix jet lag. And jet lag really happens because there is an automatic rhythm in our body which oscillates in a day-night cycle. And when you travel long distances where the day-night cycle is switched, now you're out of sync with the day-night cycle and you get jet lag. And there are ways to get back in sync that allows you to fix it. So this whole picture looks a little complicated, but I'll walk you through the basic idea. It's extremely simple. The bottom line is that there is a day-night cycle to life, right? As you go in time, every day you have a day and then night, day and night, and so on. And proteins in our cells respond to that, okay? And all that allows is sets up an oscillatory circuit where certain proteins go up and down in sync with that cycle, okay? And all that you're seeing in that slide can be simplified like this. Something gets made, gets out, then something comes back in and starts doing more stuff. And in between is delay. This feedback action has delay in it. So that means you start making a lot, then after a while you come back and inhibit, then you start making again, after a while you come back and inhibit. That's all that's going on, but it's happening in this interesting way. There are two key proteins called timeless and period. They're both related to time, as you could imagine, or tim and per. Let's say these two genes get made, follow the outer circle first, outer loop first. You get mRNA made, then the protein is made. This protein usually gets degraded in response to light. So let's say daytime it gets degraded. If not, when it accumulates to a high enough level, you get this dimerization that's formed with enough of them. With a certain delay, they'll dissociate, then they'll go back into the nucleus and inhibit their corresponding expression. So that's all that's happening. You're making a lot of protein, get out there, and if you're not degraded, you accumulate high enough, go in, start inhibiting. Okay? So this sort of arrangement, you can think of it as negative feedback loop plus a delay. And that leads to an interesting property of oscillation. And we know that just these things are sufficient for oscillation because you can recreate this circuit artificially. So consider this circuit here where you have, let's say, repressor A, B, and C. And it's set up such that there's a binding site for C in front of the repressor A protein. And there's a binding site for A in front of the gene for B, binding site for B in front of the gene for C. So the whole thing is arranged as a circular loop with just a few things in between. So if you were to look at how these proteins are getting made across time, you'll see a graph like this. Follow any one color, it goes up, comes back down, goes up, comes back down, and so on. And it's interdigitated with these various other colors, right? So if you measured any one protein experimentally after setting this up, you can, let's say, set up this gene construct, put it inside E. coli, and then watch what happens in E. coli by measuring each of these proteins. You will actually experimentally see them cycle up and down. Okay? So oscillatory circuits are crucial. They occur in circadian rhythms in us and are responsible largely for us getting jet lag when that goes out of whack. And the key components are negative feedback loops and delay. Okay? Delay is essential. If it's immediate negative feedback, then you don't have enough accumulation and it's a flat line. Okay, so that's it on circuits for now. So the first thing we talked about is memory mechanisms, and the second thing we talked about are complex ways in which genes can be put together. And we stopped with just a few simple ideas. One is positive feedback loop, and the second one is oscillatory circuit with individual examples. The next thing we'll talk about is the process of DNA methylation and how that's used for regulation in eukaryotes. Remember, during the DNA repair lecture, I briefly mentioned that 5-methylcytosine is found in eukaryotes, and it's used for gene regulation. So here's a refresher. This is where there is about 3% of 5-methylcytosine in vertebrate DNA. And if that underwent a hydroxylation reaction or a deamination, then you end up getting thymine there. And thymine, although it's a native base, would be found opposite a G in this case, and therefore will be repaired using mismatch repair. Okay? So this is taking you back a couple lectures, but I hope it comes back fresh now. Let's take a closer look at what's going on here. You have methylation that's being used as for regulation, which means you have 5-methylcytosines that accumulate. So what? Well, there's a really cool consequence of just this process happening in embryos, which leads to some unique properties of promoters in eukaryotes. So we'll talk about what that is. As a second reminder, I should tell you that this will lead to a huge change in the base pair. And also as a refresher from last lecture, remember gene regulatory proteins are binding base pair DNA without unzipping the DNA. So the major groove surface that's presented gets drastically changed if you have methylation at that position. Okay. All right, so the methylation that happens is a memory mechanism, as I mentioned at the beginning of the lecture. And it's a memory mechanism because there are maintenance methyltransferases. What these proteins can do is recognize if one of the strands of DNA is methylated and then put on a methylation mark on the other strand of DNA. So imagine you're starting with two regions that are typically CPG or CG locations. One of them is unmethylated at the cytosine, and the other is methylated. So both sides are methylated. Now you get DNA replication. And when that's replicated, you will end up, because of the semi-conservative replication of DNA, one of those strands has that single methyl, but the other, the new strand that's in orange, does not have it. So both these things are not methylated, right? The maintenance methylation mechanisms are such that they only methylate the DNA that's heavy methylated or half methylated. So there are already two locations here, right? So this CG has no methyl mark on it, and so the maintenance methyltransferase does not act here. But the subsequent CG had half of it methylated, so the maintenance methyltransferase will methylate the other half. And that's how the methylation in DNA gets remembered. Okay, so that's a mechanism by which the gene regulatory mark of DNA methylation gets remembered. Not only that, if you had epigenetic regulation or regulation that's above the level of DNA, it's often arranged in layers, and one layer can recruit the other. What I mean by layer is the level of histones versus the level of DNA methylation versus the level of, let's say, large-scale condensation of chromatin, and so on. So what's illustrated here is how one layer of regulation can recruit the other layer. So by now you should know that there are histone modifying enzymes, which are writer enzymes, and then other enzymes that can read the fact that the histone has been modified. And if you have a reader-writer enzyme, what happens is it sees the histones modified, modifies the neighboring histone, and again recruits another one that sees that it's modified, modifies the neighboring histone, and so on. So that leads to a large-scale condensation of DNA, which is recognized by DNA methyltransferases that put these little dots you can see in the loop on the DNA. So that leads to, you can think of it as a really crunching up of DNA into a compact ball, if you will, and that is repressive, meaning that does not allow gene expression to go through, and this rate of repression can be incredible. About 1 billion-fold repression can be achieved in eukaryotes by using such mechanisms. So histone methylation is the first layer, recruited the second layer, which is DNA methylation, and together they can lead to about 1 billion-fold reduction in gene expression. Okay? So these ways of regulating are really, really dramatic and can be remembered across cell divisions. So with that background on DNA methylation, we'll talk about this cool aspect of what happens to even the sequences of eukaryotes because of DNA methylation and how it gets repaired. So in eukaryotes, in all of our genes, there's this idea that if you sequenced it, you find a whole bunch of CGs near the promoter. It's called CG islands or CPG islands. The P just stands for the phosphate in between the C and the G. But you don't find as many CGs everywhere else. Okay? So the question is, how come it's this way? And we can understand why this happens if we really think about how repair works and what's the efficiency of different repair mechanisms. That seems odd. It's as though the sequence in DNA is dictated by rate of repair that happens at different sequences. Okay? So I don't know if any of you have already figured it out, but I'll walk you through this slowly. It's an important question. Okay. So let's think about a sequence, let's say that is CG, 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 and then this is methyl CG, and then CG. Okay? So there's other stuff in between, but it's all CG, CG, then there's a methyl CG, and so on. Let's say over time, 1,000 generations go by, and those deamination reactions are happening, right? So remember that the deamination of this will make this, let's say, UG, okay? And let's say this is CG, and let's say here again a deamination happened, that's UG. This would become TG, right? Because of deamination. And this, let's say, remains CG. So now UG can be recognized by DNA glycosylases and then repaired through base excision or through homology-based repair, right? But T is a natural base that's found in DNA. So what happens is that is not repaired using DNA glycosylases and is instead repaired using mismatch repair. Okay? So it turns out that repairing this is inefficient. And this is repaired efficiently. So what that means is over time, every time you have a methyl G, you repair it inefficiently and all the methylated CGs will keep changing and they'll change into TG. The only place where you won't be changing the sequence will be wherever there's CG and it's not methylated. Okay? So what that means is on vertebrate DNA, over many years of evolution, you'll end up with these black tick marks, which are the CG sequences, being located only in the promoter, because everywhere else in the embryo you get methylation. So this is how you end up having CG sequences or CPG sequences at the promoter. So what I said is summarized again here in writing. You have the deamination reaction, which leads to T, and the crux of the reason for why this happens is mismatch repair is less efficient than homology-based or base excision repair. Okay? Is this clear for everyone? Any questions on this so far? Okay, good. All right, so hope you understood that clearly. Might be important from the exam point of view. So the other thing that this methylation allows cells to do in eukaryotes is distinguish maternal from paternal DNA. And this is one way in which you're getting expression from only one copy and not from both copies. A second way in which you get expression from one copy and not both copies is the X inactivation that we've talked about. While X inactivation is going to be random, for imprinting, it's always only one of the copies. It's either maternal or paternal. It's not random. So that's a bizarre thing, because at each generation, you're getting DNA from the mother, you're getting DNA from the father, and then if you are male, then all of that DNA is now male DNA for the next generation. And if you're female, all of that DNA is female for the next generation. So how is that orchestrated? That's orchestrated using controlled DNA methylation. We don't know all the details yet, but we know some. So imagine here's the female mouse and the male mouse, and there's a gene copy that's orange or yellow. Let's say the orange is inherited from the father, and it starts out in a somatic cell, meaning in a body cell. There's expression only from the yellow. There's no expression from the father's copy because it's methylated. Okay? Now, when you have a female mouse during embryonic development or during gamete development in the production of eggs, all these methylation marks get removed, making it effectively like both copies are now female. Whereas in the male, you get imprinting where you have methylation mark in all copies. Okay, both the paternally or maternally acquired. So that in the next generation now, the same thing gets reconstituted irrespective of what the DNA is, what the color is. See, if this is orange or yellow, it doesn't matter. It only matters where it came from. If it came from the mother, it gets expressed. If it came from the father, it is not expressed. Okay? So this form of regulation is really important in us, and in fact the insulin growth factor receptor is one of the most famous imprinted genes, and it controls how much we grow, really, in response to growth factors. And how this whole thing is established isn't clear, but the crux of the answer is there's methylation in the male, and unmethylated DNA in the female that gets established again and again at each generation. Okay? Now, why bother with methylation? What could that do for you? I hope you heard about barrier elements and insulator elements in the previous lecture. That's where this comes in. Insulators and barriers are kind of boundary elements that prevent gene regulation from spreading across genes. If you have an insulator element, let's say it's insulating enhancers from the gene, it's usually insulated with the help of a protein that binds. You don't need to know the name of the protein, CTCF, which prevents this enhancer from activating here. But in the paternally inherited chromosome, you have the methylation that happens, which then allows this enhancer to activate this gene. So in this case, methylation is allowing expression, but in other cases, you can have methylation not allowing expression. It just depends where the regulation is happening. Okay? Right. Okay, so we come to the next way in which organisms are able to express from only one copy of the DNA and not the other, and that's called X chromosome inactivation. How many of you have heard of the calico cat before this? Maybe some of you have a calico cat. Okay? So they're pretty cool things, and they have that color because the coat color gene is located on the X chromosome, and you end up having this really mottled, patchy appearance because there are clones of cells that are inactivating or activating coat color together. Okay? Clones of cells, meaning you start out with a one-cell embryo and you're dividing, you're making about, say, 1,000 cells, and at that time you decide I'm going to inactivate one or the other X chromosome, and based on what the color is on the two, you will get all descendants of that particular cell having that color, and all descendants of other cells having a different color. So that's illustrated here, say a paternal X and a maternal X. Then one X at random. Notice that unlike in printing, here any one X gets at random inactivated, but only one gets inactivated. And all subsequent divisions have only that inactivated. So for example, here the paternal is inactivated. It's shown here sort of crunched up. And all its descendants have the paternal inactivated. And on the right, the maternal is inactivated. All its descendants have the maternal inactivated. How this process happens is also still under really active investigation, but we know some really cool hints about how this is happening, and it has to do with RNA. Okay? So there is an RNA called exist, and there are actually other complicated RNAs as well called T6, which is the reverse of that, and so on. But you don't need to know all those details. Suffice to say, there's an RNA called exist, which is made from the X inactivation center, or XIC, and it then has this property that it can spread all along a chromosome, but it spreads only in cis, meaning only on the same chromosome, not on the opposite X. So you can imagine where if the X level is high enough for it to start spreading, there's some other mechanism that prevents it from happening on the other chromosome, and you have the spreading in cis to generate this inactive X chromosome. Okay? That RNA is leading to the condensation of the chromosome so that now you don't have gene regulatory proteins being bound and you don't have any activation. Okay. So it's transcribed from the X inactivation center. Now this whole process of inactivating an entire chromosome is part of a more general mechanism for equalizing the dosage of DNA between males and females. Males and females have different levels of DNA, and they have different levels of DNA in different ways in different organisms. For example, in mice, you have XY versus XX, just like in us. But in flies, you have XY and XX. In C. elegans, you have XX and versus a single X. There's no Y chromosome. And in some other organisms, like birds, you have even more different things. So, but in all cases, there's an equalization mechanism that exists. Okay? The whole point of this inactivation is to make sure that the male X content and the female X content are the same. So one whole chromosome is inactivated so that these two have the same X. But you can achieve that in a different way as well, and that's used in Drosophila, where instead of one of the X being inactivated in the female, you have the one X in the male being upregulated. Okay. And in C. elegans, it's yet another way in which it's done. You have both X's being downregulated by about 50%. It's bizarre how this whole thing is achieved across the whole chromosome, and it's still an area of active study, but we know that that's what happens. Okay? You get a downregulation. So the general reason for why this happens is to equalize the level of gene expression from the X chromosome between males and females. And this is called dosage compensation. So that brings us to gene regulation and the many different steps along the way. I think we'll discuss 8 steps. So I'll pause here and ask you any questions so far on what we've talked about: memory mechanisms, circuits, and imprinting and X inactivation. Any questions? Okay. All right. Hope you guys have this clear. There may be questions on this in your exam on Tuesday. So there are many more steps than the first step of expression, and as I said, first step of deciding to transcribe RNA, and as I said, all those steps can be regulated. And in what follows, I think we'll walk through a single example for each of these forms of regulation. So the easiest way to think about it is just think of the remainder of the lecture as just marching along this continuum of you've decided to make RNA. From then on, at what step can you regulate and how are you regulating? And we'll only discuss one specific example going forward. Elongation, splicing, 3' end, etc. So we begin with elongation. So let's say a RNA polymerase sits on DNA and starts transcribing. The rate at which it elongates and whether it elongates all the way through can be regulated. And what is shown here is one example of that elongation. And I'll use this to introduce what is really a relatively recent discovery of classes of RNAs called ribose switches. As you can imagine, this does the switch type activity like RAS or any small GTPase protein, turn something on or off, but it's entirely made of RNA. And it's found in bacteria very commonly. And let's say it's upstream of a gene that's required for purine biosynthesis, purines G and A. And it might form a structure like that. And when there is nothing to bind, the polymerase will go. Remember, this is the transcribed RNA that's folding back on itself and forming a structure. It forms that structure because that structure can bind a G. And it can bind a G with an incredible number of contacts. This is now RNA binding a small molecule, not protein binding a small molecule, but RNA. And these are all hydrogen bonds indicated, and this is showing the specific interaction of G. You don't need to know these interactions. I won't be asking you to draw any of these things. But I want you to know that RNA itself can form a structural pocket in which G would fit. Now, if G is bound, what happens is downstream there's a change in structure. So if you go back a couple slides, you'll see that downstream there wasn't any base pairing here, but when G is bound, you create the stem loop, and then usually a run of U's. So in bacteria, if you have a stem loop and a run of U's, that's a signal for termination, and the polymerase stops and gets kicked off. Okay? So this is a way in which the elongation of the polymerase is regulated by what small molecule gets added. Okay. So guanine stabilizes the downstream transcription domain. The second thing is splicing. So we just discussed elongation, and the second thing is splicing. Splicing is still a very active field of research, and we really don't know how alternative splicing happens. And alternative splicing refers to the many different ways in which you can splice the same thing. So you have a gene, let's say yellow schematizes introns and blue schematizes exons. You could have alternative splicing in all of these ways. These are really sort of common sense ways. If you just look at it and think about what are the many ways in which you could include or exclude an intron. You might decide to skip an exon, so that's an optional exon kind of alternative splicing. Or you might not have an intron and just translate through. If there are no stop codons, or if the reading frame did not change, that's fine. You'll make a little bit more of a protein, but you'll keep going through. Mutually exclusive exons, where you either include this exon or include this exon. Or an intron in which you can now use bits and pieces of the intron, which would be through using an internal splicer. So you should be able to tell if you were asked, show a couple of ways in which alternative splicing might happen. Remember that both options, having the 1 and the 2, together define one alternative form of splicing. You could either do this or that. And then the second one would be another of these 1 and 2 options. So this alternative splicing really allows organisms to dramatically increase the diversity of proteins they can make. So the count of genes for organisms like us is in the order of 20,000 or 30,000. Okay, it's still changing. But that's the order of number of genes that we have in total. But look at this one protein. It's called DSCAM, or Down syndrome cell adhesion molecule. It's found in the Down syndrome critical region. But it has so many alternative splice forms that are possible that you can get 38,000 different forms of this protein. Okay. That's more than twice the number of genes that are found in Drosophila. Okay. So that's how many different forms you can have. And what we know about its function is that it's required for neuronal development, and it guides different neurons binding to each other. So you can imagine it's like having thousands of different kinds of specific glue that would specifically bind to each other based on which exons got used. So that's a dramatic enhancement of diversity that this particular protein goes through simply because of alternative splicing. And entire processes can be controlled by just alternative splicing. And alternative splicing is so pervasive that, for example, in Drosophila, all of sex determination happens through alternative splicing. I think that's coming up in the next slide. So before thinking about a specific example, you can think of negative control and positive control. We've talked about this in detail before in the context of lac operon and other operons in bacteria. Negative control would be where having the repressor prevents splicing. So yellow is again intron. Normally you have splicing, but if you have this repressor, you now don't splice. Positive control is where having this protein, an activator of splicing, allows you to splice. Okay? And both these forms get used in Drosophila sex determination. So remember I said that this whole X inactivation process, or changing the dosage of X, is only to make sure that expression is the same in male and female. And how that's really controlled is by taking a ratio of the X chromosome to the autosome set. Yes, organisms can do math. They count the number of X chromosomes, count the number of autosomes through mechanisms that people are still figuring out. And based on that ratio, regulate downstream genes. Again, you don't need to know the names of these genes. They're all related to sex, so it's sex lethal, transformer, double sex. They together give you either a male fly or a female fly based on activity. But they are arranged and regulated purely by alternative splicing. Forget the words for now, just look at what's here as the two gene schematics. You have yellow introns, you have those light blue alternatively used exons, and on the left, looks like there's one form of splicing going on, and on the right you have proteins like these circles here, green circles, that are being used to change the kind of splicing that happens. Okay? So I'll let you read this at your leisure, but the bottom line idea is very simple. On the left, you have non-functional proteins that are made because every time you make such splicing sequences, you get stop codons or something that leads to degradation of the RNA, and you don't get functional protein. But on the right, you begin by first making functional sex-lethal protein, which changes its own splicing and affects the splicing of the downstream gene or transformer. And then that makes functional transformer protein, which binds with another one and changes what you make, such that in the end, now you're left with two different kinds of double-sex proteins in the female versus the male. Okay? So alternative splicing has a cascade of events that just leads to the production of two different proteins that regulates male development versus female development. The third thing is forming the 3' end. So you're transcribing as you're making this long RNA, you're removing bits and pieces, that's splicing. And then at some point you have to stop. So based on where you stop, you'll make proteins that are either really long or really short. And this is used in our immune system, where you have antibodies that are found membrane-bound on cells or antibodies that are secreted. So the secreted antibodies are shorter than the antibodies that are anchored to the membrane. And the ones that are anchored to the membrane have a transmembrane domain at the end, a hydrophobic peptide at the end. And how that's regulated is shown here. You either make this long transcript, which has a stop codon inside the intron and a stop codon that is after this. And if the first one gets spliced, you end here, and then you get this long piece. But if you end up not going all the way to the end in transcription, then no problem. You stop prematurely. You make a very short bit here that's shown here. That's hydrophilic. And it's within this other intron. So by switching between these two, let's say a B cell or something is activated. Now you want to make a lot of antibodies that are secreted. Then you switch from making this membrane-bound and start making the secreted version. Okay? Now, number 4 is editing, which is what goes counter to central dogma, if you will. You go from DNA to RNA to protein, and the information is thought to be precisely preserved before we discover editing. And editing is done by a class of enzymes called adenosine deaminases that act on RNA, or ADARs. What these do is they typically recognize some sort of stem loop structure in an mRNA and then convert adenosine to inosine, which is an A to I reaction, which is a deamination reaction. That's why it's a deaminase. And that's shown here. You have the amino group that's removed, and this is inosine. Now this changes the base pairing properties dramatically. An A would base pair with a T, but an I would can base pair with a C. So imagine if this is happening on an mRNA, you'll have anticodon from the tRNA base pairing differently, so that now you have a different amino acid being incorporated into the protein. And in fact, in us, in humans, there is one particular protein which leads to, in which if you don't have this ADAR, you go from glutamate to arginine. You don't need to know that specific change, but you end up then getting seizures because this channel now misfunctions. So this is not an obscure thing that happens, but it's really used by organisms as a form of regulation, and it's been selected for in some cases. Okay, so editing can happen along the way. So if you've made the RNA, the first thing that you need to do to get protein from it is to get it out of the nucleus, because translation is happening in the cytosol. Okay? We can discuss many different examples of how this happens, but for now we'll just focus on the HIV example, because the HIV virus, if it, let's say it's latent and integrated in the genome, and it needs to get its entire genomic RNA out, it's faced with this challenge. The long RNA, which is its genome, the HIV has an RNA genome, has no splicing in it, but it needs to get out of the nucleus. Okay? So how does HIV get out of the nucleus can be used to as an illustrative example to understand the regulation at this level. You don't need to know all these protein names, but one protein in particular will feature in this process that's called the Rev protein, and it binds a specific region on the long genomic RNA called the Rev response element. That's what RRE stands for. Okay? So just remember this, the Rev is located somewhere on the right, the early genes are on the left, the late genes are on the right, meaning the early genes get made first, the late genes get made later. And normally in a cell, most of the RNA is obliterated inside the nucleus, very little gets out. So only about 1/20th of the transcribed RNA gets out. Most of it gets obliterated because they're not spliced. So imagine if this is an integrated viral DNA inside a cell, you make this long unspliced RNA, it'll be immediately subject to nuclear degradation. But if you make these spliced small RNAs, they can get out. Eventually, at some point, you'll make these Rev RNAs, okay? That'll make the Rev protein. And once that Rev protein is made, that can then go back into the nucleus. And it has this property of binding to the Rev response element and protecting unspliced long RNA from degradation. Not only that, it helps the unspliced RNA to get out of the nucleus, and now the entire virus can assemble in the cytosol. Okay? So the crux of the matter is you have a protein called Rev, which needs to get made. It's a late protein. Then it goes back into the nucleus, protects the long RNA, ships it out, and now HIV can infect the next cell. So that brings us to getting RNA out into the cytosol. Now we've got it out of the nucleus into the cytosol, but in the cytosol, again, there are forms of regulation. In some cells, this form of regulation can be really dramatic and really important, especially in neurons. We have some neurons that go from our dorsal nerve cord all the way to the tips of our toes. That's an enormous distance for molecules to travel. So you can imagine all sorts of RNAs are needed at the tips of the cell, so there must be transport mechanisms. You may want to put RNAs in different places. So how is all that done? We can think of it as three specific mechanisms, one of which is directed transports. That is, you deliberately move the RNA from one place to another. A good example of it is in neurons, where you want to take the RNA from the nucleus where it's getting made all the way out to the periphery where the axon ends. A second one is where you allow random diffusion of the RNA, just moves about freely, but then there are some capture stations, if you will, which are essentially proteins that can bind the RNA that are localized in specific places. So the RNA is moving about freely in the cytosol. If it bumps into that protein capture station, it's stuck. Okay? So that's a second way to regulate. A third way would be the RNA comes out and it keeps getting degraded at a certain rate. All the time it's getting degraded, but except when it's bound by these capture stations. Okay? So those would be three different ways in which you can have regulation of RNA localization. And here's an experiment that shows how that happens, and we now know that a lot of this regulatory information of where does an RNA stick is found in the 3' untranslated region of RNA. So if you took an mRNA with an intact 3' UTR, it might localize precisely to this outer edge of a Drosophila embryo. In blue are just nuclei that are found on the outer edge. But if you lose the 3' UTR and you mutate it, then you find it in this cytosol, inappropriately localized in the general germplasm. So that brings us to, you've got the RNA out of the cytosol, you make sure it's in the right place. The next thing that happens is the ribosome has to get on there and translate. So here again we have many complicated things that can happen, but I'll show you, I think, 3 or 4 examples which are all exactly the same thing and easy to understand. And they're bacterial examples. In bacteria, ribosome entry occurs through binding to the Shine-Dalgarno sequences. I hope all of you have heard of that term, Shine-Dalgarno sequence, yes? Yes? Okay, good. So you've heard of that. That's where you have recruitment of the ribosome, and so you can imagine by regulating the rate at which ribosome can be recruited there, you can regulate the rate of translation. So one example is, say you mask ribosome getting there using a protein. That would be the example shown here. Or you change the base paired structure, which prevents the Shine-Delgarno sequence from being seen, using some other input from the outside, like temperature, which just simply opens up the orange Shine-Delgarno sequence. That's using the environment. Or you might use a metabolite, like that example of guanosine binding for the riboswitch. You could have another small molecule that binds and changes the structure of the RNA so that now the ribosome is bound up, the Shine-Delgarno sequence is bound up. Or finally, you could use some other RNA to base pair to this Shine-Delgarno sequence so that the ribosomal RNA can no longer bind this Shine-Delgarno sequence. So all these are just different examples of the same idea, whether you capture the Shine-Delgarno sequence and sequester it or not. Okay. In eukaryotes, the situation is more complicated, as you might imagine. And we'll talk about a few different aspects of it. One is where there is a sequestration of the initiation factor IF2. So EIF2 is eukaryotic initiation factor. Here the initiation is for initiation of translation. It turns out it is a GTP binding protein that's typically bound by GDP, and you have guanine nucleotide exchange factors which will switch GDP to GTP and make it active. Now if this EIF2 is phosphorylated, then it can act like a dominant negative. It'll just bind up the other protein, that is the exchange factor, not let go, and all the other EIF2s will be inactive. Okay? So the basic idea is, instead of directly having a sequence in bacteria where the ribosome binds, you regulate the initiation factor, whatever the protein is that's required to start translation, which is the eukaryotic initiation factor 2. And the translation happens not on linear RNAs, and this is from a few lectures ago, where you know that if you look at translating RNA, it's really kind of a circle. There are proteins that bind the poly A tail on the mRNA, and there are proteins that bind the cap, the 5' cap, and they interact with each other, as shown here. And this is the structure which is competent for the ribosome to get on and translate all the way through. Okay? Now, viruses can hijack this idea by using what are called internal ribosome entry sites, or irises. All that they do is they mimic this structure. They don't have a 5' cap, and yet they can bind this EIF4G protein, which will bind this poly A binding protein, making this circle. So that now regulates the entry of ribosome. So far we've seen two different ways. If you phosphorylate the EIF2, you prevent ribosomal entry. If you mimic the circularized RNA, you trick the ribosome into entry. And that's what viruses do to translate their protein. The final one, I think, is degradation of the RNA. So every time an RNA is made, or really any protein is made in a cell, or any molecule is made in a cell, it's always a competition between whether it's being kept stable or it's being degraded. There's something degrading everything in a cell, literally. You can think of it that way. And in eukaryotes, the point of adenylation at the end, and 3' end, and having a cap at the 5' end, is to protect the RNA from degradation. Because the degradation happens through nucleases that like to chew from one free end, called exonucleases. So they like to chew from a free end. So two ways in which you destabilize the protein are, one, you might shorten this poly A tail so that the chewing can get into the message and you have degradation. Or if you cut the message in half, the 5' half now has no tail, that gets degraded rapidly. The 3' half has no cap, and that gets degraded rapidly as well. So always for every message, it is a balance between, is it getting translated by having the cap being bound appropriately by the cap binding protein, forming this productive circle, or there is an enzyme called deadinylase, as the name suggests. It removes adenyl groups, or removes the poly A tail, and that leads to degradation. And for every message in a cell, it's a competition between those two. So to finish up, I'll give you one complex situation which suggests that what happens to an mRNA in response to exactly the same event can be dictated by what the context is. And this is found in the case of iron regulation in us. Iron is a trace metal, but if you have too much of it in a cell, it's toxic. So its levels have to be regulated really carefully. So there are two conditions we can think of, whether you're starved for iron or you have excess iron. Okay? And in both these cases, there's regulation at the level of RNA stability that's happening. And that's dictated by whether another protein that binds iron binds to the RNA or not. And there are two RNAs we'll talk about. One is ferritin, which is just an iron-binding protein that's found inside cells. And the other is transferrin, as the name suggests. It's a protein that transports iron across cells. Now, both of them have the structure where there's a stem loop that's found on one end or the other. On the left is ferritin, on the right is transferrin. And they're bound by some protein called a conatase. You don't really need to know the name of the protein. Some protein just binds both of them. Now, what happens when you don't have iron is that you have different regulation in both sides. On the left, you have ferritin is getting not made, but transferrin is getting made. So if you don't have iron, transferrin is made so that more iron can come in. But when you have iron, the same thing happens in both places. The conatase protein is kicked off, but the outcome is now the opposite. Now ferritin is made because you have a lot of iron. The intracellular protein that binds iron, which is ferritin, is getting made, but the transferrin, which brings in iron, is not getting made. Okay? So the same events can lead to different outcomes based on the context in which the regulation happens. So I'll stop with that, and only up to today's lecture will be in your exam. I'll send out a notice saying